The ventricles of the brain have the following divisions: two lateral ventricles, a third ventricle, and a fourth ventricle which communicates with the central canal of the spinal medulla. Lateral ventricles. The two lateral ventricles have the following divisions. Number 1, anterior horn which projects into the frontal lobe. Number 2, a posterior horn which projects into the occipital lobe. Number 3, inferior horn which projects into the temporal lobe. The anterior horn has a roof made up of the corpus callosum, and the lateral wall is made up of the caudate nucleus. It projects into the body of a lateral ventricle at the trigone of a lateral ventricle. The trigone of a lateral ventricle communicates with the body, the occipital horn, and the temporal horn of the lateral ventricle. The temporal horn has a medial wall made up of the hippocampus. Its dorsolateral extreme is formed by the amygdaloid nuclear complex. The occipital horn has a roof made up of the tapetum. The lateral ventricles communicate with the third ventricle by the interventricular foramina. The third ventricle has a lateral wall made up of the thalamus. It communicates with the midbrain which surrounds the cerebral aqueduct. This aqueduct links with the fourth ventricle. At the rostral extreme of the medulla, the fourth ventricle opens into the two lateral apertures called the foramina of Luschka. Meninges. The meninges are the coverings of the structures of the central nervous system, that is, the spinal cord and the brain. The meninges are usually divided into cranial and spinal meninges, depending on whether they cover the brain or the spinal cord. They can be further divided into three parts, three types, or three coverings. The most external is the dura mater. This is followed by the arachnoid mater and then the pia mater. The dura is also called the Pachy meninges. It is the thickest of all the coverings of the neuraxis. It is divided into two portions, the more superficial part of which is attached to the cranial periosteum. This is the cranial dura. The cranial dura is followed by a deeper dura directly covering the brain substance. The space that separates the dura and the arachnoid mater is the subarachnoid space. The subdural space. This space is important clinically because of the possibility of hemorrhage as a result of a tear in the branches of the arteries within the space. This type of hemorrhage is known as subdural hemorrhage. Hemorrhage may also occur clinically into the space above the dura by a tear in the branches of the meningeal arteries that supply the dura itself. It is then called epidural hemorrhage. The arachnoid and the pia are thin and are applied to the surface of the brain very closely. They are together called the pia arachnoid complex or the leptomeninges. This leptomeninges is very thin, but there is also a potential space between the two maters called the subarachnoid space. Cerebrospinal Fluid Csf is contained within the subarachnoid space. This space also contains subarachnoid granulations which allow for the flow of CSF in a unidirectional fashion. The CSF flows through the arachnoid granulations into the main bloodstream by entering intravenous sinuses around the brain. The subarachnoid space communicates with the ventricular system of the brain through two lateral foramina of Luschka and one median foramen of Meninges. CSF therefore moves freely within the ventricular system, communicating with the subarachnoid space. Hemorrhage may occur in the subarachnoid space, and this is then called subarachnoid hemorrhage. Cisterns are spaces containing large amounts of CSF placed between the pia arachnoid complex and the dura. The largest cistern in the brain is the cisterna magna, placed between the cerebellum and the medulla oblongata. This is the cerebellomedullary cistern. pontine cistern is placed anteriorly between the two halves of the pons. The superior cistern is placed around the posterolateral part of the midbrain. Other cisterns are the interpeduncular cistern around the cerebral peduncle, chiasmatic cistern around the optic chiasm, and the lumbar cistern in the spinal cord. These cisterns are important because they can be used in outlining the brain in radiological imaging and in pneumoencephalography.